Hello boys and girls, this is Mr. So. Welcome back for another lesson of biology. Today we are going through lesson 28 of chapter 27, the gene mutation. Before you start watching the video, remember to like and subscribe my channel and also share the video to your classmates. Let's start. One of the very famous mutation events that happens in this century would be the nuclear accident occurred in Fukushima, Japan, Fukuoka in the 2011. In year 2011, a very strong earthquake occurred near Japan's sea, and this leads to the destruction of the nuclear facility in Fukushima, Japan. Because of the destruction of the nuclear facility, this led to the leakage of some very strong radiation from the nuclear plant. This radiation leads to mutation and further leads to, for example, people working in Fukushima having cancer, 例如血癌啦 we call this the blood cancer or leukemia. Not only does the human living in the Fukushima being affected by the strong radiation and leading to the mutation, some of the organisms such as plants living in Fukushima also change their phenotype, their looks, such as the flowers shown in here. They are very special on their look. 輻射異形化啦，令人恐慌嘅變種菊花。So as you can see, the strong radiation leads to mutation 突變 in the genes or in the DNA of the living organisms and change their phenotypes, their looks. Another example of mutation in Fukushima include a formation of very weird antenna, 觸鬚啦 in the butterfly in Fukushima after the leakage of radiation. So obviously mutation 突變 it leads to a change in our phenotype, in our character, the looks. But how exactly does mutation affects our phenotype? It is actually changing our DNA in a sudden and permanent way. And this change can be separated into two subtypes. They could be a gene mutation or a chromosome mutation. Let's take a look at the first type, gene mutation first. Gene mutation refers to the change in the base sequence, the base A, T, C, G of the DNA in gene. And the change in base sequence can occur in 4 different ways, such as deletion, insertion, substitution, and inversion. Let's take a look at them one by one. Let's take a look at the base sequence encoding strand in a normal person. It has the base with triplet code ATG, CAT, GTA, and TTG, which codes for these 4 amino acids. However, let's observe together if you have gene mutations such as deletion, you delete a base C, what happens? If you delete the base C, the host sequence shifts to the left for 1 base. And if you have an insertion, the host sequence shifts to the right for 1 base. And this forms totally different amino acids after the point of mutation. The deleting of base, like without the C, or inserting of a base, like adding a base G, will lead to a complete new set of triplet code formed. And we call this situation a shifting of reading frame of the DNA. And after the point of mutation, which is located in here, the insertion point or the deletion point, all the amino acid sequence afterwards will be altered. So this is how you change the reading frame, shifting the reading frame, leading to a change in amino acid sequence after a point of mutation by deletion or insertion of base. The third type of gene mutation is called substitution, such as substituting or changing a base C to base T. The last type of gene mutation we call it inversion. That means inverting the position of 2 bases. For example, from CA to AC. And these 2 types of gene mutation, substitution and inversion, it only changes one triplet code and leading to only one change of amino acid. As you can see, substitution and inversion, it only changes one triplet code, and therefore it will only change one amino acid. Let's do a question to check your understanding on gene mutation. Explain why a base deletion can have a much more significant effect in terms of the final protein produced than a base substitution. In order to answer the question, let's take a look at the case happened in substitution. Are the triplet codes affected? How many triplet codes are affected? How many amino acids are affected? Let's write it down. In base substitution, how many triplet codes are affected? Let's take a look. The only triplet code affected is the second triplet code. So therefore, in base substitution, only one triplet code is affected. Or you can say it's altered, it's changed. Which leads to a change in one amino acid. Alright, but let's take a look at the case of deletion. When you have a deletion of base, what happened? The deletion of base change how many triplet codes? It changes all the triplet code after the point of mutation. And after changing the triplet code, all the amino acid after the point of mutation, they are also changed. So therefore, in a base deletion, the reading frame of DNA is shifted. Which now leads to a change in the amino acid sequence after the point of mutation. And at the end, this is going to affect the formation of protein. If you have a different amino acid sequence, a different polypeptide, this is going to affect the folding to form a protein with three-dimensional structures. So therefore, base deletion have a lot more effect in terms of the formation of protein than the base substitution. So overall in here, you have 4 marks. For the first mark, you tell me in base substitution, only one triplet code is altered and changing one amino acid as 1 mark. And in base deletion, the reading frame of the DNA is shifted, 2nd mark, and leads to a change in amino acid sequence. after the point of mutation, the third mark. And this changing of amino acid sequence affects the folding to form a three-dimensional structured protein. You get the fourth mark, the last mark. Please go to your textbook page 18 to do the MCQ. That's it for the video of gene mutation part 1. I will see you in the next video. Bye bye.